We're going to split this remaining part of this chapter into 3 lectures. This one will be viral classification, and the next lecture will be viral reproduction, and then finally we'll review some plant and animal viruses and also some unusual sort of infectious agents called prions and viroids. Previously, we had talked about viral morphology and saying that each virus has a nucleic acid core, and it's either DNA or RNA, but not both. The DNA could be double-stranded or it could be single-stranded, and the same thing with RNA, it could be single-stranded or double-stranded. Some other aspects of the genome, it's either DNA or RNA, as we've said, single-stranded or double-stranded, but the DNA or RNA could appear as circular, as we see in bacteria, or it could be linear, like we see in eukaryotes. It could just be a single polymer of DNA or RNA, or it could be multiple segments. In influenza, there's 6 to 8 RNA pieces of RNA. Obviously, the genome is going to be smaller than the host cell's genome. The DNA is going to direct the host cell to make new copies of the virus, and RNA viruses are going to be directly transcribed and replicated inside the host cell. We've also seen that there's a protein capsid that surrounds the nucleic acid core. It's made up of smaller units called capsomeres. And then there are, in some viruses, a third layer called the envelope, which is like the cell membrane, like the host. One of the most common ways that viruses are classified today is using what's called the Baltimore classification. It's named after David Baltimore, who is a Nobel Prize-winning virologist who developed the classification system. You've got morphology of the capsid and then differences in genetics, but primarily the focus is on how the messenger RNA is produced during the replicative phase of the virus. So we're going to go through each of these groups and see how they're classified. So the Baltimore system is separating the viruses into groups. The group 1 viruses, of which herpes simplex is an example, these are viruses that are double-stranded DNA viruses, and their messenger RNA is produced by transcription in much the same way we see in the cell itself, using the host cell's enzymes. Double-stranded DNA is used to code for messenger RNA that then produces the capsomeres for the virus. The group 2 viruses are single-stranded DNA viruses. In this case, they convert their single-stranded genomes into double-stranded DNA after they've infected the cell, and then transcription of the messenger RNA can occur from that, again using the host cell's enzymes. The group 3 viruses are double-stranded RNA viruses. The strands separate, and one of them is used to make the messenger RNA using RNA-dependent RNA polymerases that are encoded by the viral genome itself. The group 4 viruses are single-stranded RNA viruses with what we call positive polarity, which means that the genomic RNA can serve directly as the messenger RNA. In order to make more copies, then intermediates of double-stranded RNA called replicative intermediates are made so that then the genome can be copied into the new viral particles being produced. Group 5 viruses are also single-stranded RNA, but now the polarity is negative polarity, meaning that the sequence is complementary to the messenger RNA. As with the group 4 viruses, the double-stranded RNA intermediates are produced, and then from this, the messenger RNA is produced, and also the negative strand genome is converted from the double-stranded RNA. The group 6 viruses are diploid, meaning they have 2 copies of single-stranded RNA. But now as the virus enters the host cell, it's going to go through what's called reverse transcriptase, which means that the RNA is going to be used to make DNA, double-stranded DNA. The DNA then goes to the nucleus of the host cell and inserts itself into the host genome. This is HIV, the human immunodeficiency virus. From that point on, then, like the central dogma we saw in Biology 1, the messenger RNA is produced by transcription, and new viral genomes are produced by transcription. So that is the Baltimore system. Are double-stranded DNA viruses. The groups that then make single-stranded of DNA to act as messenger RNA in each, and then these are converted back to double-stranded using reverse transcriptase to produce the new viral genomes.